Welcome back, everybody, for the afternoon session. I'll just invite Dr. Mary Phillips to come up and share this session of early career investigators presenting.
Thanks, Ben. And again, thank you very much, everyone, for being here this afternoon. I know some people will be coming back after a lovely lunch and post presentations. So it's my pleasure this afternoon to chair what I think is going to be an incredibly exciting session of presentations based on the theme of bipolar disorder, looking at various different techniques, neuroimaging, genetics, behavioral. And we're going to have 3 presentations. Each presentation is going to be about 10 minutes or so. I can be a bit lenient. And then we can have 10 minutes for Q&A. And I think the idea, Ben, is to wrap up within about an hour or so. But we can have a bit of discussion. And also, if there's time, perhaps have room for a bigger discussion at the end of the 3 talks to tie themes together. So, as I say, we have 3 presentations this afternoon. It's my pleasure, first of all, to present the first speaker, who is Dr. Clement Zai, who's going to be talking on neuroimaging, genetic risk, and biomarkers of bipolar disorder. So let me just tell you a little bit about Clement. Dr. Zai, he's a staff scientist at here, an associate professor of psychiatry at the university here. And his research interests, as you can tell from the title, are very wide-ranging, including genetics, pharmacogenetics of psychiatric phenotypes, bipolar disorder, schizophrenia, PTSD, and suicide. And I think this talk is going to be incredibly exciting because he's going to be tying the neuroimaging genetic risk together. So thank you very much, Dr. Zai. I can't wait to hear your talk. Thank you.
Thank you. Thank you for coming, and thank you for picking this abstract for an oral presentation. I'm going to talk about polygenic risk scores and suicide-related phenotypes, outcomes in bipolar disorder and high-risk youth. And these are my disclosures. And these are the learning objectives: to describe the genetic component of suicide and also to understand polygenic risk score analysis in the context of youth bipolar disorder. And to introduce, there are over 700,000 deaths due to suicide worldwide each year, with 4,500 of them in Canada alone. And with each suicide death, there are at least 20 suicide attempts. And having a psychiatric disorder is a major risk factor for suicide. And it's especially true for youth bipolar disorder population, where 30% of them actually have a history of a suicide attempt. So studying this population is particularly important. Now, family, twin, and adoption studies have supported a genetic component of suicide, and the risk of a suicide can be explained approximately up to 43% to be genetic. And genome-wide association studies, or GWAS for short, it's an approach that offers screening single nucleotide polymorphisms, or SNPs for short, across the genome, across the human genome, to identify novel associations. And this approach has led to sort of the discovery of genetic associations in complex traits like BMI and height, as well as risk for complex diseases like heart diseases and bipolar disorder. As for suicide, the Manhattan plot on the right here, it shows the results of a GWAS of suicide attempt on over 43,000 suicide attempt cases and over 900,000 non-suicidal controls. And on the x-axis are the chromosomes, and the y-axis are the p-values in a negative log 10 scale, and with each dot representing the association finding for each SNP across the genome. And the GWAS here actually found 12 regions in the human genome with genome-wide or study-wide significant results with suicide attempt, as shown by the dots above the red line. And this GWAS also indicate that the effect of each of these SNPs are very, very small. So one way of actually looking at genetic analysis in suicide is to look at combining these effects together in a polygenic risk score. And this sort of a series of diagrams or figures that shows how polygenic risk scores are calculated. But first, polygenic risk score is defined as a measure of disease risk due to either the significant SNPs or even just the tested SNPs in a GWAS. And in order to do that, we need GWAS study in sample A. In our case, it would be a GWAS of suicide attempt in the 43,000 suicide attempt cases and the 900,000 non-suicidal controls. And with each SNP, there will be an effect, a different effect on the risk of suicide attempt. And because we want to take a look at how these genetic risk of suicide might affect sort of an outcome or some phenotype in a different sample. In our case, sample B, which would be a youth bipolar disorder, for example. And for each individual in sample B, there could be a different number of risk alleles or risk variants across these tested SNPs, as shown by these blue and white dots. And each of these dots or SNPs also have a different effect, as I mentioned. So the polygenic risk score, or PRS for short, is essentially a summary of the sum of all these, the number of these risk alleles, and then weighted by the effect of these SNPs on suicide attempt. And so because of this, then we can actually genotype sample B for these SNPs and then get their polygenic risk scores for suicide attempt. And then this leads to a distribution of these scores. So there would be people who have high scores who would be considered to have high genetic risk for suicide attempt, and then those who are low risk of suicide attempt would have the low polygenic risk score for suicide attempt. And using these scores, we can actually test out how, if they are actually associated, these scores are associated with suicide outcomes. And in the literature, PRS for suicide attempt have been associated with suicide attempt in youth, and also similarly, PRS for like major depressive disorder has also been associated with suicidal ideation and suicide attempt in general youth population samples. But polygenic risk studies in bipolar and high-risk youth are lacking at the moment. And so we set out to actually test the hypothesis that the genetic risk of suicide attempt given by polygenic risk scores are associated with the risk of suicide attempt in our youth bipolar and high-risk sample. And our sample has, it's collected from the Center for Youth Bipolar Disorder in Toronto, and it's age 13 to 20, and they were administered the KSAT PL, and where 151 were diagnosed with a bipolar disorder, while 149 were considered to be high risk due to their familial loading and high risk of major depressive disorder and psychiatric comorbidity without actually meeting the criteria for bipolar disorder. And they were also given the adolescent longitudinal follow-up interview to collect data on suicide attempt, non-suicidal self-injury, and suicidal ideation. And also they were also data on family history of suicide attempt were also collected. As for genetic data, these were collected using standard methods, using the Illumina global screening array, and also standard quality control and whole genome imputation were carried out at CAMH in-house. As for the method for calculating PRS, we use the PRS with continuous shrinkage using the default parameters. And we calculated these PRSs based on summary statistics from GWASs of schizophrenia, bipolar disorder, major depressive disorder, ADHD, suicide attempt, and risk tolerance based on their sort of previous studies with of suicide and these PRSs. And we ran logistic regression models and using dependent variables of self-harm, suicide attempt, and suicidal ideation. As for independent variables, we look at each PRS individually in a standardized scale. And we included the sex, population, structure, and diagnostic groups, as well as family history of suicide attempt as covariates. And these are the results. Overall, females are more likely to have self-harm or suicidal ideation than males. And also the bipolar group have almost twice the risk of suicidal ideation than the high-risk group. And as for PRS, The MDD-PRS seems to be higher in youth with suicidal ideation compared to youth without suicidal ideation in our group, as shown in this violin plot. But our PRS for suicide attempt was not associated with the status of suicide attempt in our sample. And so a summary of our results is that, as mentioned, suicide attempt PRS was not associated with suicide outcomes in our sample. But recently we actually found that the bipolar PRS was associated bipolar diagnosis in the youth, in our youth sample. And so it may suggest a potentially different genetic component of suicide in the youth bipolar compared to like adult samples or other populations. And also, we also found the MDD-PRS to be associated with suicidal ideation, which agrees with the previous studies in youth. And some limitations, moderate sample size is usually the case for genetic studies, while our PRS also did not consider a lot of other variants within the genome, including rare variants, non-SNPs, complex variants, sex chromosomes, and mitochondrial genome. And on top of that, the risk of suicide attempt in this case, based on the SNPs found in that GWAS, it's around 5%. So our GWAS, our PRS is actually capturing just a small portion of the risk, even the genetic risk of suicide attempt. So a lot of work still needs to be done. And finally, some future work is needed, including like a follow-up assessment that may capture additional suicidality cases, and also consideration for other factors like childhood trauma, psychosis, socioeconomic status, and substance use. And larger samples could also enable additional analyses. And these are the acknowledgement. Dr. Goldstein and Dr. Michele Bertocci from the Center for Youth Bipolar Disorder for their support throughout the study, and Dr. Trevor Young and Dr. Jim Kennedy for their input as well. So thank you.
Thank you very much. Very interesting talk. Are there any questions from the audience? Because I know I have a couple, but I wondered if anybody had a question to start off with. Okay, well, may I ask the first question then? So you didn't replicate the findings about suicide attempt, PGRS. Can you speculate a little bit more about why that was the case? You're saying that perhaps you need another risk score for children with bipolar disorder rather than reliant on adult scores. Could you just sort of elaborate a bit more about why perhaps you didn't get the finding?
Yes, so partly could be, as mentioned, the potentially different genetic component of suicide in this particular population. Another is maybe it's not the suicide attempt PRS may not be precise enough, so we may need to sort of combine perhaps additional factors to bring out that association if there's any hidden.
Yeah. And related to that, can you say a little bit more about the actual genes that go into the polygenic risk score? So I'm thinking mechanism here. So okay, so you didn't replicate the attempt, but the risk for bipolar disorder was there, and also the risk for suicidal ideation you did replicate. So I noticed that one of the genes you showed earlier on was a dopaminergic function gene. Obviously, I was noticing that. So I'm just curious to know as to, I know it's a big question, but for me, GWAS is great, and the polygenic risk scores are really great for detecting risk. But can you say a little bit more about what it might tell us about mechanism to help us in the future? Sorry about the question.
No, it's good. Actually, it's sort of, in a way, there could be some criticism actually in terms of PRS in this sense, in that it's crude because you're just looking at all the SNPs that are past a certain significance level. So one way of looking at this is like the D2 receptor. So it's been associated with a lot of different phenotypes, including schizophrenia and substance use, sure. So there are sort of developing methods to look into including functionality into PRS. And in some cases, we can actually look at maybe overlap between bipolar disorder and suicide attempt. And then looking at further, looking at the functional effect or the pathways within those, and then developing more pathway-based polygenic risk scores. Yeah. So that may be a way to actually answer the questions.
Yeah.
Thank you.
Thank you. Are there any other questions? on this, because I think you sort of exemplify the sort of how the fields can meet together. You know,
Yeah.
the field of genetics could perhaps talk a little bit more and the field of neuroimaging could also talk a little bit more with the field of genetics to try and understand those pathways. But that's great. Thank you very much. I think there's going to be time for questions at the end, and we can perhaps bring some strands together. But that's
Thank you.
very great talk. Thank you. Thank you. So we're right on time, which is great. It's my pleasure to introduce the next speaker, Dr. Michele Bertocci, who actually works in Pittsburgh, and I've known Michele for several years, and she was instrumental actually behind some of the work that I presented, particularly the ketogenic diet work that I presented earlier. Michele is interested, she's a guru at many things, but I would say Michele is basically uses a lot of multimodal imaging, as you're going to hear about fMRI, but also spectroscopy, and is particularly interested in using these techniques to characterize psychiatric risk. And her talk today is neuromarkers of mania that distinguish inpatient adolescents with bipolar disorder from those with other psychopathology. Thanks, Michele.
Do I just keep going? I don't know how to get to. Do I just keep, okay. Okay. Thank you. It's an honor to be here. Thanks for having me for World Bipolar Day. I'm Michele Bertocci. So I'm going to tell you the learning objectives for my talk. I'm going to talk about, give you a familiarity with the INCABS imaging study, and I'm going to describe two methods that have shown potential to be used to distinguish bipolar disorder from other similar diagnostic groups like ADHD and depression. One's a neuroimaging study and the other is an actigraphy study. And so these might be useful for risk calculation. So the inpatient child and adolescent bipolar spectrum imaging study, which is a mouthful, is a 5-year NIH grant that recruits from the INCABS unit, which is a specialized bipolar spectrum unit for child and adolescents at Western Psychiatric. And I think it's the only one in the world, which kind of speaks to some of the issues we were talking about earlier, that we don't care about bipolar disorder in children or don't think it's important. So on this unit, there are 13 to 17-year-olds who are a referred sample from the psychiatric emergency services so it's the emergency room at Western Psych. And all of these kids who are referred to this unit have some inkling of mania type behaviors at the time of being in the hospital. But the overall aim of the study is to be able to differentiate people who have bipolar disorder from other difficult to distinguish disorders. And we're trying to use both multimodal methods and multimodal data collection types. So we have a training sample, and that is children on the unit who have bipolar, frank bipolar disorder types 1 or type 2. And then the other group is called other psychopathologies. So this group of kids have not bipolar disorder. So they could have depression, they could have anxiety disorder, they could have ADHD, they could have disruptive behavior disorders, but they have an absence of any lifetime mania type behaviors. And this is discovered once they have been on the unit for several days. So this is our training sample. And then we have a testing sample of people who have children who have bipolar unspecified, other specified. So these kids have some subthreshold mania or hypomania, and we expect about 30% of them to convert to bipolar in the next 2 years. So we will use the results from our training sample to see if we can predict which of those kids will convert to bipolar disorder. So that's the aim of the study. I'm going to first, the study elements are all completed while they're on the inpatient unit. So their research staff do a case ads interview with both the child and the guardian. And then Rasim Diller, who is the psychiatrist, he's the MPI in the study with me. He has trained the research staff in case ads interviewing, and he also does the consensus with the interviewers. We have multi-day mood and energy self-reports while they're on the unit. They also wear an actigraph while they're on the unit for as long as they can tolerate it. We have multi-day observations. So the kids, the research staff go and observe the inpatients while they're on the unit, making sure they're wearing their watches and completing their scales. They have an fMRI once they are stable enough to go to the scanning facility, and they complete the MRI there. And we have neurocognitive assessments, which they also do once they're stable enough. And then we follow them for 2 weeks post-discharge, doing mood and feeling self-reports, and they wear the actigraph for 2 weeks. And then we follow them again for 2 years to see who is going to convert to bipolar disorder. So for the neuroimaging sample, I'm going to be talking about a card guessing monetary reward task, which is similar to what Mary talked about earlier. And we had 38 inpatient bipolar 1 or 2 who were able to complete the scan. We had 60 kids with other psychopathology and 24 healthy controls. They were the hardest to recruit, still are the hardest to recruit, the healthy controls, I guess because of COVID, I don't know, but we've had trouble recruiting them. So the image you see is a whole brain main effective task. And we're seeing that we are, in fact, seeing the reward network being activated to this task across all of our participants. But what we're really interested in the study is being able to differentiate our groups. So can we say, is there neural activity that differentiates bipolar from other psychopathology specifically? And we used a reward mask, a pretty extensive reward network mask. And we show that there are subcortical differences across our 3 groups. So the bipolar participants had reduced activity in the left amygdala and the bilateral thalamus to this reward expectancy. And it's a pretty high effect size, 0.5. And it's across all 4 of these regions. We're speculating that the reason for this reduced activity in these subcortical regions, which may not have been predicted by some, that it's because this task isn't activating this reward network enough in these people. So the children who have bipolar disorder tend to be risk-taking, as we all know, or impulsive, and this just wasn't interesting enough for them to care about. So they have reduced activity. They need more stimulation in order to activate these regions. That's a hypothesis or speculation. But due to the nature of our recruitment, we wanted to see how this neural activity related to symptom measures. And we have a variety of measures that we record from our kids. So we use the brief mania rating scale to look at mania, obviously. The mood and feelings questionnaire for depression, the positive and negative affect scale for we use the PANIS, and for anxiety, we use the SCARED. And we show that there's no relationship between these 4 neural regions, so left to reward, left amygdala and bilateral thalamus for depression, anxiety, or positive or negative affect. But we do predict mania scores, both past month and lifetime, using these 4 neural regions. So this potentially is a useful, could be useful in a risk calculator for determining the difference between bipolar disorder and other psychopathologies. Another potential useful metric is activity. So we know that kids differ when we are looking at kids who have disorders. Part of the diagnosis for bipolar disorder or depression or ADHD is differences in daytime activity. So we're going to be looking at historical actigraphy data. So this is chart review from inpatients on this in-cabs unit. And this work was done by Farzan Varhafard. He's a postdoc in my lab. And so we know using standard ANOVAs, we can look at the difference between, we can tell the differences between different groups. So there are certainly differences between bipolar with ADHD and bipolar without ADHD in their daytime activity. But we can't tell the difference between clinically challenging measures participants to distinguish. So for instance, bipolar with ADHD is very difficult to clinically differentiate from ADHD without bipolar disorder. They're often misdiagnosed. And also bipolar without ADHD and other diagnoses like depression, they're difficult to diagnose. And we can't do even with very large samples, we're not seeing those differences using ANOVAs, using standard actigraphy or just standard ANOVA measures. So Farzan wanted to use, he has experience with artificial intelligence and wanted to look at seeing if we can differentiate or classify groups with difficult to distinguish pathologies using this historical chart review data. So we have inpatients from 2014 and to 2023, and they have those diagnoses as you see. And just part of standard clinical care, they were wearing the actigraph if they were able to on the unit. Racine would just have them wear the actigraph while they were on the unit. And we range from 61 ADHD without bipolar to other diagnoses, which was our largest group, that 127 kids were able to wear the actigraph for at least 4 days. And on average, they wore the actigraph between 12 days and 16 days while they were on the unit. So we have a large amount of data to use for these AI models. And in addition to the daily activity that I'll explain to you in a minute, we had age and sex at birth and also time of day when these activity measures occurred. So the approach Racine took, or sorry, Farzan took, was to create 4 hourly bins of non-overlapping hours of maximum activity and minimum activity over the course of the day. And that's excluding nighttime. So there's a standard bed at 10 o'clock, wake up at 7 o'clock on the unit. So those hours were excluded from the day from this analysis. But so we're just looking at the daytime hours or the awake time hours while they're on the unit. We also looked at differences between the max and min, and then also engineered activity. So things like moving average, and these are standard measures to look at longitudinal data. So moving average removes the outliers and looks at the trends across the time series, and then rolling mean and the lag. Rolling mean is looking at 3 days, averaging over 3 days, looking at how the trends change over those 3 days meaned, and then the lags looking at the days, 1 day prior and 2 day prior. And what Farzan showed, he tested 4 models: a random forest model without feature selection, a random forest model with feature selection, XGBoost with and without feature selection. And the worst model was the random forest without feature selection, but we're still explaining or able to accurately classify 77% of the groups, which is pretty good. And the best model was the XGBoost with feature selection, and we were able to correctly classify 92% of the groups. I'm going to apologize for the color problem here. So on the left-hand image, the XGBoost with feature selection is in red, and on the right, it's in that mustard color. So just pointing that out. But even if you look across different groups, we were able to accurately classify between 82 and 95% of the groups of these very challenging to distinguish psychopathologies. And I think the 82% for the bipolar without ADHD just really speaks to the heterogeneity of bipolar disorder generally. And the features that were the most important for this analysis were, we had age and sex at birth. And then the other features that were most important were all moving average features. So looking at the trends across maximum and minimum daily activity in these inpatients. And even in this very standardized setting, that, you know, they're on the unit, they can't really leave, they can go to the gym, but their activities are limited because they're on the unit. But this suggests that even on this very standardized environment, we can still see differences that classify these groups pretty effectively, which may indicate some biological difference that we can actually measure. Future studies should really look at outpatient actigraphy and outpatient samples and see if we can classify those groups. So again, this is another metric that might be useful in risk calculating or in classification and being better able to diagnose our groups. I'd like to thank Rasim Diller, who's my MPI on the study, and then all our staff and postdocs, and of course, Mary and Boris, who are our mentors who have been very supportive over the course of our careers. That's it.
Thank you, Michele. Thank you, Michele. That was great. And, you know, good example of multimodal approaches to a very, very difficult clinical question. Were there any sort of questions for Michele about any of the sort of the data that she presented? I have a couple of questions, but well, let me start off. First of all, obviously, I've got to ask about the reward task, the neuroimaging task. I know you expected that. And so your findings were obviously subcortical amygdala, and just as a basic question, and those were lower in the children with the bipolar spectrum. Can you speculate as to, you know, what might be happening in the cortex? I know we talked about this in the past. Perhaps, you know, taking dimensional approaches, looking at mania risk across all the groups together. I mean, can you just have some thoughts about what might be going on there in terms of what's going on in the other parts of the reward system that might predict mania risk? Yes,
thank you for the question. I think part of it has the reason we are not, one reason that we may not be saying ventrolateral prefrontal cortex or other prefrontal regions, maybe because of the nature of the groups. So our other psychopathology group could have people who have bipolar, could have ADHD or conduct types disorders, which both have impulsivity related to them. So this may be kind of washing out the prefrontal regions that we expected to see. So potentially looking at a more dimensional approach to mania specifically may show us those regions more. Well, I haven't done that analysis, so I can't actually say too much more than that.
Thanks. And I did also have a question, if I may, about the activity findings, which are fascinating. You know, can you say a little bit more about which specific aspects of activity really were distinguishing the groups?
Yes. Well, let me show you the graph.
Yes, I couldn't quite read all the x-axis very well.
So I'm going to go over there and point to them so you can see them. So the most important was age in months. And this is the color we're looking at, which is the XG boost with feature selection. It's very comforting to see that they are similar across all measurements. For all of the each of the measured models that we used. But we're looking at, so max fourth means the max 1 is the most active that they've been during the day. So fourth is the kind of in the middle of the day. And then we're looking again at all of these are moving average. So we're looking at trends across the time series instead of the individual measures themselves. So because the individual measures have a lot of noise associated with them, they also have a lot of outliers that may be contributing to differences. So most of them are, we're showing max and min moving average, and we're also showing the differences between the max and min moving averages that could potentially that are contributing to the differences we see in these groups. So.
And do you think that these are going to relate generalized to the outpatient world? Because this is obviously the inpatient world, and as you said yourself, it's very restricted. It looks as if it's a very convenient measure.
Yes, easy to collect. Yeah. People don't mind wearing them. I have one that is not a clinical one, but I do wear it all the time. Probably most of us do. I don't. So we do test to look at within and outside of how well these results will generalize. And that's actually one of the measures that we get from the output. which is, sorry, I have to go back a couple. So recall and also talks about how well we could potentially be using this in another sample that is similar. Now that's an inpatient sample, obviously. So I don't know how well it would reflect in kids in the wild, but I do think it's worth trying and worth testing. So.
Thank you. Were there any other questions at this stage? As I said, hopefully we'll have a few minutes for discussion at the end. So, well, if not, thank you very much, Michele. That was a lovely short insight into some of the lovely work you're doing. Thank you very much.
Thank you.
So our third and final speaker in this afternoon session is Dr. Bradley Macintosh. And Dr. Macintosh's title is Multimodal Deep Learning for Accurate Diagnosis in Bipolar Disorder, a Proof of Principle. Now, I'm just going to say a few words. I'm sure many of you know Dr. Macintosh. Dr. Macintosh is a professor of medical biophysics here, and basically does a lot of work about neuroimaging research and clinical translation, and has made huge contributions to the centre's work over the last decade. And it's a really great pleasure to welcome him back here again today. Thank you.
Thanks so much. It's a great session and great day. Feeling very lucky to participate. And the one thing I didn't realize, the truth or dare, this is Ben saying, I dare you to present on a topic that's not bipolar disorder on World Bipolar Day. So I have to apologize from the outset. This is a priming of the pump. You'll see the reason why we started with and not a bipolar sample, but an autism spectrum disorder. So it's great to get a nice setup here from Michele. I'm going to give an executive summary so you know what the next couple minutes are going to be about. So open source data is a wonderful way to start to think about neuroimaging methods and use of deep learning. So that's where we pivot to the ABIDE consortium, which is focused on autism spectrum disorder. So if things work nicely in a cohort where there's a very large sample, then it gives us a lot of appreciation for when we move to maybe a CYBD sample. And there's many AI approaches, so we're going to consider two. And we're going to do sort of like control experiments, where we're going to try to predict age or sex. And then the question is, where do we go from here? So actually, it was a really great setup. This is a very complex slide. And the good news is that Michele teed it off, and we also saw some stuff on receiver operator characteristics, thanks to Eric. So you really don't have to pay attention to anything on this slide except for a few things. So if I had an MRI data set, could I take all the beautiful information, intensity values in the MRI data and just simply churn it through, decoding what's in the MRI data and produce an estimate? Is this next case a patient or is it a control? So image-based classification, that's what we're doing here. And among all the different options, AI is a concept, machine learning is the thing in practice, deep learning is hot. So that's how we got into deep learning based on the classic convolution neural networks. So we're going to try to do classification. It behooves me to remind you that there's like decades of work to get us to where we are today. And in 2016 is another big jump up, because you could take your data in a machine learning framework and it could be like an accordion, that you'd have this huge amounts of features in your deep learning network, and you can just squeeze it and squeeze it and squeeze it, which is essentially what the DenseNet was designed to do. It's worked really well in other contexts. It has a good chance in imaging as well. So, and then of course you can see some of the usual suspects like ChatGPT on this narrative. So the objective is, if you simply had an MRI data set, could you classify individuals as being, in this case, ASD, yes or no? And we predict that more you feed the neural network and train on it, the better the classification ought to be. So we're going to have two candidate markers. It's going to be an anatomical scan, and then we'll do functional scan. We're going to see, is there an opportunity to classify individuals? So we need a large sample to do this because you need to train on a lot of data. And in fact, in this case, from this open source initiative, there's 1,000 that were eligible. And then I should mention that this work was done in collaboration with Ali Jahani, and he has no disclosures, neither do I. Should have said that first. And as we moved through and created a cohort, we made what we think is a very good sample. It's going to be perfect number of ASD and controls, 351. They all have MRIs. So we're going to focus on the T1, and we're going to focus on the functional, the resting state fMRI scan. And you can see that this is where it becomes of interest because in the context of neurodevelopmental changes in the brain, the average age is 15 years old. In this case, it's skewed towards more males, but there's no difference in the demographic details between these two groups. The other challenge and richness of this is that the data is coming from 17 different sites. So we know that if we're going to be making a guess, this individual is ASD, yes or no, there's going to be lots of sources of variability, and we're going to embrace that. So to kick things off, and thanks again, Michele, for helping, we're going to use XGBoost, which is great because it uses tabular data. And so you have a spreadsheet, rich spreadsheet, very long and hopefully very wide as well. You can just simply pass that in. The XGBoost is a great framework to perform classification based on tabular data. But I'm more into computational radiology these days. So the DenseNet model is the deep learning model. It's actually going to take the entire stack, the full 3D volume, each person, and every possible brain voxel has an opportunity to contribute to the classification. So we get into this rich feature space. It amounts to, I'm going to ballpark around 5 million trainable parameters in our giant deep learning calculator. And the experiment 1A and 1B is the control test. So in individuals that are free of a mood disorder, or in this case ASD, can we simply predict whether they're male or female, and whether they are median split older or younger? Experiment 2 is the one that we actually are really interested in, because we're going to do the diagnostic classification, ASD, yes or no. But we're going to prime the pump and consider XGBoost versus DenseNet first. So on your left is the DenseNet results. On the right is XGBoost. This is the classic confusion matrix. So if your model is doing the right job, then you will only see values along the diagonal. So the blue, you can see in this case, the DenseNet had an accuracy of 81%. And this was only using the anatomical scan. So if you have an anatomical scan, you can say, is this a male or a female? I don't know, a radiologist in the room, can they tell? Sometimes you can tell sex differences from the scan. So now this was done in what's called like a K-fold cross-fold validation. So we had different ways of churning through the data to give us a mean value. So 18.9 individuals were correctly assigned to be male, similar number for female. But you can see it gets it wrong a few times. That's the way it is. With XGBoost, the performance is similar, but a little bit lower in this particular case. The XGBoost, the way we pursued that is we chopped the brain regions based on standardized atlas, and then produced the gray matter value estimate within each of those regions of interest. So it amounted to like 200 regions or so. Now here is predicting whether the individual is above the median age or below. So the average, this is trying to guess, is this individual above 14.4? And here in the controls only, and the accuracy values are comparable now. DenseNet seems to do better than XGBoost. So frank disclosure, we were going to do the DenseNet anyways. And that's all we did in the next experiment. So the next experiment is using, again, the full MRI stack goes in. And the cleverness of this is that you can actually stack your dense net. So there's 3 million trainable parameters, and then you load in your next data, and it also is looking for the features, trying to crack that code. What's in the MRI data that I can squeeze like an accordion and put out a single binary estimate. So the data here is, I'll just walk quickly here. So anatomical, functional, anatomical plus functional. So that's how you can see that there is a gradual performance improvement when you use the anatomical plus the functional data. And then on the right-hand side is, as I mentioned to you, the 10 folds of cycling through the data, producing the receiver operator characteristic curve each time. And the average area under the curve in this case is 0.84. So I think the accuracy value here, the highest accuracy was achieved when you use the resting state functional data plus the structural data. So I think that bodes well. And now in summary, a relatively large sample of children and adolescents were used in this classification task. And the 3D DenseNet had some face validity as providing the means to produce an objective estimate. Is this individual ASD, yes or no? So it's the type of decision support that I think is encouraging. Our control tests of age and sex classification, they were pretty good in the scoring above 80s. And it was kind of reassuring, since we're all here planting seeds and growing together, that if we are using multiple sources of data, seems to be a theme, both the anatomical and the functional, then that produced the highest accuracy for this classification. And I truly believe this is a great wide open area for research in psychiatry. Now we just have to start sharing, finding ways to share the data and make a case that these research MRIs have clinical value. And I think with that, I'll highlight some of the references and thank the co-authors for their amazing work in doing this. This is an offshoot project that just started a couple of months ago. And with that, my sincere thanks to the Center for Youth Bipolar Disorder, and happy to have lots of discussion.
Thank you very much, Bradley. That was really, really, impressive. And, we are open to thinking about other disorders that's other than bipolar disorder. We love everything, but that was very good. And I think it's a good example of how techniques can be applied to many different disorders. So, first of all, were there any questions from the audience? Yes, Michele.
So I think XGBoost is more efficient for longitudinal data. So it kind of, this may be a more comment than a question. So maybe it makes sense that it does not as well.
Yeah, I'm repeating the question. So trying to reconcile XGBoost had slightly lower performance. I think XGBoost is great when you have a lot of complex data, like kind of what you were demonstrating, and how you would be able to capitalize on different features from data, the different calculations. But just simply reporting like a gray matter estimate in zone 1, 2, 3, 4, 5. I think we kind of knew that it's probably not the best, showcasing the best the XGBoost can do. And in fact, I was kind of back my way out of that and say, why would you touch the data and start playing around with it? Just put in the actual raw data. And that's where image-based classification we had to do a couple of things to the data, especially the fMRI, but really don't touch the data. Just put it straight in and train in the most native format for it. I think that's my way of saying the DenseNet has its place. Yeah.
Sorry. What were your thoughts about multiple scanners? Were there scanner effects? Were there?
Well, the question was there any investigation into potential side effects? And I guess that's where, when it comes to, we could look at that with the XGBoost, but with the DenseNet, it's just like la 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 la. We have no idea. So the fact that it works across 17 sites, I think that kind of bodes well. And I'm really interested in site effects. So maybe there's a way to tweak, find ways to normalize and boost the performance further. Yeah.
Great. So if I may, I have a question. Really going back to a similar question I asked Michele, can you say a little bit more about which specific features went into the classifier, that helped with the differentiation?
Okay, so when it comes to the dense net, we really have no idea, right? So it's that's the truth. I mean, it's a black box. That's the one downside to deep learning is in these particular approaches is that the explainable AI people sometimes talk about is that if you want to do explainable AI, then it's a slightly different experiment. So I would love to say more about that. But in this particular case, I really cannot say. In the case of XGBoost, when you get your sort of salience features, the feature selection, you get to see which brain regions were moving the needle in the classification. So that's where they were really good. And it's nice to meet. We have a lot of common interests here. So I think that's where you compare it off, depending on, do you want to be able to explain the result, or do you just want to just brute force it? And in the context of radiology, maybe one day, there's lots of clinical MRs around. You just say, it's okay, just keep feeding this beast. We have lots of data, lots of research questions. So hopefully I answered your question. Yeah.
Thank you. Yes, Ben.
Is there explanatory effects of structure and function? Does that overlap for individuals?
Oh, yikes. That's a tough one. Well, outside of this, we know that there's a lot of kind of shared variants. There's certain brain regions where there'll be reductions or increases in brain structure. And then it seems as though there's possibly a compensatory effect. So I think you could design the experiment to sort of try to pit one of the imaging measures against the other and then test to see which one is able to, do more, have better prediction or classification accuracies. But I think at this point, the real interesting thing is that you don't care. The fact that the two scans, if you have two scans, it's not going to hurt you. If we saw the performance was dropping by adding the functional scan, then that would be a way of saying that it's sort of polluting the system. But that we didn't see that actually. It's just sort of charting upwards. So of course, we would love to slide in the cerebral blood flow scan and see things keep moving up and to the right. That would be ideally what we would see. And task fMRI as well would be cool.
Yeah. Any other questions specific? Yeah.
Brad, I had a question about the fMRI in the dense net. You said you put in the belt maps instead of the raw resting state. How did you choose the belt map over the raw or some other processing of that fMRI?
Yeah, that's a great question. So the simple way to answer that is that although the neural networks and the computation has gotten really good, and we were using high performance computing environment, there's absolutely no way that you can load all the fMRI data. At least I don't know how to do it. Maybe Google can figure it out for us. But so it's too many volumes. So we'd have to chop it down. We'd have to find a way to reduce it. And that's a really good point. There's additional information in the fMRI data that we could consider as a few more channels. Good idea.
Any other questions? I'm just seeing if there are any in the chat. Not specifically, but Brandy, that was absolutely great. Thank you. And perhaps I think Ben, have we got time for 5 minutes for perhaps a general discussion, drawing together some themes, perhaps? I don't know. I don't want to put you on the spot, but Michele and Clement, if you wanted to come up so we can, have sorry. So I do not put people ever on the spot. That's not my style. But I think what we've seen today is some really great talks and synergy. Starting with Clement and then Michele and then Bradley, and I guess before I move on, there was a question for the first speaker, Clement, which I missed. It came through later in the chat. And perhaps this can kick it all off because I had some questions that were similar. And the question is, are there other PRS that you could use that might relate more to like impulsivity, so are there other PRS measures that you could use related to things other than diagnosis, etc., or suicide? So for example, impulsivity that actually might get closer to home.
Yeah, thank you. Thank you for the question.
So you can answer it to everyone.
Oh, okay. So we use sort of the risk tolerance as a sort of a proxy for impulsivity in our case. Because I guess there isn't actually large enough GWAS of impulsivity that can actually generate a PRS for impulsivity that's powerful enough, I guess, to be used for PRS analysis. So risk tolerance at this point. Hopefully in the future, actually, if we have like a GWAS of impulsivity measure in the population data set, then may be able to do more. Yeah.
So we really need more genetic markers
More
of
data, yeah.
dimensions that might precede the diagnosis or things like suicide. Yeah, and I guess please take
Yeah.
do you want to sit
Oh,
there?
right.
Otherwise, I'll fill your chair.
This one.
So I guess that brings me to a nice segue into the question I had for all speakers, which was, you know, you all have wonderful work and complementary work. But to me, it seems as if there could be sort of transdiagnostic approaches using dimensions, so like impulsivity. So Michele, I mean, obviously we talked about that, and my question was, you know, are there dimensions of pathology that you could use to relate neuroimaging and the motion work to and the activity work? And kind of the question was already asked. And then Bradley, I guess for you, you're looking at diagnostics, but could you use, I mean, presumably you've been thinking about using the AI to, I don't know, discriminate along a spectrum of pathology, for example, which might be more sensitive. And did you, I mean, perhaps if I could ask you each to think about ways, or perhaps you're already doing this, I'm sure, but perhaps tell us more about how you're addressing this question, because it relates very much to the RDoC, but before that, you know, it relates to dimensions of behavior that we know really underpin the diagnosis that we're really interested in.
So
I'm sorry, you know, perhaps it can people hear everyone? I'm thinking of people online, so I put the microphone over there.
yeah, so the goal of our study is classification for better, so that we're better able to diagnose people and treat individual diagnoses. But if we're looking at risk, then we would potentially looking at impulsivity as a risk factor, that we could look across diagnoses that that present with impulsive behaviors. And also there's also mania and depression and anxiety. All of these are dimensions that we could potentially examine. But again, our study specifically is about classification so that we can treat people in a more efficient way and I think the fact that Racine has a very low recidivism rate for the unit, I think it's under 10% of the kids come back to the emergency room and are inpatients again. And he has not had a single suicide in the 20 years that he's been the director of his unit. Speaks really well to his diagnostician ability. So, and that when people are diagnosed well and given a particular classification, they can be treated with the correct medications and followed, you know, longitudinally in order to be well. So that's, but certainly looking at dimensions is an important factor.
Mhm. Yeah, it's a great question. And for me, when I think of the work that we were seeing here, classification, it helps to just turn it into a data-driven. It's not in your head, it's in the data, you know, so it gives us like really strong construct that we know what we're studying. There's, you know, the biological substrates, and you can explore with the fancy methods, which now deep learning offers that. But if I think about dimensional work, I think I'd like to go back to, you know, more simpler methods that people agree upon, that how you would construct that as risk profiles. I think it'd be better to not have too many fancy bells and whistles. I'd rather think about it as the simplest measures that many people could use, so that you're not left with sort of our ivory tower has this amazing dimensional construct and the rest of you can't do it. That to me would defeat the purpose of such an approach. So in summary, keep it simple when it seems to be the most clinically important.
Yeah.
And use the fancy stuff when you're trying to attract more attention and start to, you know, build bigger cohorts and more collaborative. That's kind of the way I see it.
Yeah. Did you want to add anything to what you said?
So I guess in terms of suicidality, because like to add to my previous, I guess, my discussion before, so there may be sort of like some unique genetic component of suicide within the youth bipolar sample, but there are also probably some factors, genetic factors that actually cut across multiple diagnoses. And this is actually what we're trying to do also in other genetic studies, to look into like impulsivity and actually measuring impulsivity among the suicide attempters or suicidal ideation sufferers. And just a sort of a whole spectrum of these suicidal behavior or suicidality individuals. And on top of that, also looking into like stress response and also aggressiveness, self-aggression.
Were there any other sort of big questions for, sorry, any other questions for the panel? Because I think we're almost getting to a close. I mean, I think, you know, what I've heard a lot about actually this last hour is harnessing AI for a start, and multimodal techniques, unbiased approaches, so heuristic approaches. I think that's what, you know, has come across very clearly. And I think that's great. I think there's a wonderful role for this, for the work, and if it can help answer clinical questions, as you were saying, Bradley, I mean, that's actually what we all hear about, right? I guess, you know, the other side of thinking about it is that to develop new treatments and new interventions, we kind of got to know what's going on somewhere biologically, so that we can say, okay, we can identify who's at risk and when we should intervene, but what intervention should we be giving right now? And I guess that's where the mechanistic work can come in and work in partnership with this wonderful big data approaches. I mean, any other thoughts on that? Because I have to say there's a big tension, isn't there, in research about, do you focus on mechanism or do you focus on efficacy for interventions? Do you focus on mechanism for understanding the sort of how, or do you understand, you know, basically, you know, when? You want to understand when and what, you know, who's at risk. So I think, don't you think we sort of need approaches that bridge the two together, so we can do both? We can identify who's at risk, who's likely to have a certain pathway diagnosis, and what's the best treatment for them. So that's the kind of not an easy thing to do here, but you know, we've got the technology to do this.
Yeah. Well, maybe one comment on that is I wear another hat where I do a lot of stroke research. Yeah. I wasn't involved in the trials where the new trials were endovascular thrombectomy, but this is a complete game changer. The efficacy was just really impressive. So do the surgery, do it fast. You can even wait. It's even within 12 hours, you can pull the clot out. But what I didn't realize is that they cherry picked in the early trials. They cherry picked the most appropriate candidates to get a good signal. And then they said, okay, as soon as we loosen the inclusion criteria, guess what happens? The effect size drops. What they knew was going to happen. So they had a good mechanism and they had a good cohort. And then honestly, it's revolutionary. You don't need to apologize for setting your trial up so that it succeeds. So I think we'll embrace that. Don't be shy, say, you know exactly who to go for and how to do the trial. So that's where I'm really inspired by the trials that you're doing, because I think you're making some concessions, but it's going to give some quality signal.
Thank you. And any other points anybody else want to make on this? Because I think all three of you are working on such cutting edge world, and it's impressive the resources and how much you can achieve, with these multimodal approaches and utilizing work that's been done before, but using it in unique ways, and that's really great to see. Any other questions at this stage? Otherwise, I think we're pretty much on time for the next break. Is that right then? 3:10. And thank you to the speakers. Wonderful work. Thank you all. Thank you for being here. That's really exciting.